Welcome, everyone, and thanks for joining in. And thanks to David and Rand for organizing this wonderful series and for inviting me. I'm looking forward. So I heard that my last name is difficult to pronounce, and so I made a little pictogram here to help you along in case you needed it. So neuroscientists often think about neurons as performing specific tasks. For example, a heartline Kupfler neuron in the retina with center surround receptive fields is thought to detect contrast in the visual scene. And Hubel-Wiesel neuron in the primary visual cortex detects oriented edges, which is all, of course, necessary to segmenting and understanding the visual scene. But when you look at biology, my impression is that the neurons are similar both on physiological and anatomical levels. Of course, there are different details, but it seems they're all kind of performing the same job. And so I'm fascinated by a question whether there is a universal algorithm that all the neurons are performing, perhaps with slight modifications, but on different inputs. And that's why they appear like they're doing different tasks. This is not a new idea, of course. In neuroscience, perhaps one of the most successful achievements of theoreticians has been the efficient coding theory, which postulates that neurons encode information they receive in the most efficient way. The exact definitions may vary, but the work started by Barlow, Laughlin, Attic, and many others led to a lot of predictions about the properties of neurons in their early sensory processing streams that have been verified experimentally. However, the efficient coding approach fails when you march deeper into the brain. It's harder to explain from that perspective what neurons do in the cortex, and it becomes close to impossible for motor neurons. So is there another way to think about what neurons do on the algorithmic level? I should, of course, mention that we are all living in the shadow of the greats, who, almost 3/4 of a century ago proposed a simple algorithmic model of a neuron based on the neuroscience data they had available. And this McCulloch-Pitts-Rosenblatt neuron is postulated to perform weighted summation of its inputs, then pass the sum through the nonlinearity and then output. This McCulloch-Pitts-Rosenblatt neuron responds online in an immediately, and is a purely feedforward device, of course. And it must be really good abstraction because, all AI nowadays is built on it. Even ChatGPT is, so it must be really good. But the point of my talk today, and this is the take-home message, is that this model is oversimplified, and not in the sense that it's missing some biological details. I think it is conceptually wrong that the correct algorithmic model of a neuron should take into account that a neuron is part of a loop and acts as a feedback controller in that loop. And that is central to understanding neuronal operation. And what I'm going to claim is that McCulloch and Pitts basically threw the baby out with the bathwater by ignoring this loop. Not their fault, of course. That was the state of neuroscience at the time. So what is this loop? Well, I think that loop should include the synaptically coupled neurons, the neighbors of this neuron we're considering, other neurons that can return signals back to the inputs of the neuron under consideration, and perhaps even the external environment. This loop has some kind of dynamics. Here, for concreteness, I'm using discrete time linear dynamical description, but that doesn't really matter. And the goal of the neuron, I'm postulating, is to produce a control signal U that influences the dynamics of the loop. And then the results of this influence are observed through the observations Y that are basically the inputs to that neuron that are used to evaluate the effectiveness of its control. So that's the basic take-home message that I want you to have as a result of this talk. And, you know, this might sound a bit surprising, but actually this control theoretical view has already been applied successfully to the brain as a whole. So instead of a neuron, I had the whole brain here, multiple people have suggested that we should think of the brain this way. But to my knowledge, no one said this about a neuron. And I think that maybe the reason is that such kind of control, even for linear dynamical systems, is actually computationally rather difficult. So although it's known how to do it, and in some cases we even have closed form solutions, they're hairy expressions involving matrix multiplications, even inversions. Just to bring this point home, let's go through the steps that the neuron has to do to successfully control a linear system like this. So first, we cannot expect, of course, that the neuron a priori knows the specific dynamics of its environment. So it has to learn it from the data, perform so-called systems identification. In this parameterization, this would amount to learning matrices A, B, and C. After that is done, the neuron would have to compute optimal control, K, based on your favorite control objective. And then you can't really implement control in terms of the observations Y if the system is only partially observed. For example, if the dimensions of Y are less than the dimensions of X, or there is noise, observation noise added, you have to control in terms of relevant state variable, which is X, that has to be inferred from the data, and that's why I call it X hat. So all those three steps, as I said, are known for the linear dynamical systems, but are computationally very difficult. And it's hard to imagine that a simple biological neuron by itself can even represent all those variables like A, B, C, and K, forget about computing them. So that's probably why this was never suggested. But 20 years ago, there was a game-changing development in control theory where they proposed a different new approach to control, which is called a direct data-driven control. And the idea there was to, for the controller to map the observations Y to the control signals U directly, bypassing the intermediate stages that I described and never explicitly representing A, B, C, or K. And the way that controller would learn the mapping from Y to U is because it has previously seen pairings of trajectories of Y and U from which it can learn the effective control. So that's what I think is happening with real biological neurons. And this is, of course, a big difference from the usual, say, error back propagation-based learning in deep learning networks, because all the relevant signals, such as the control signal U that the neuron has to produce and the teaching signal Y, are locally supplied and therefore can be learned by biologically plausible local learning rules. So I realize that this is maybe a very non-intuitive view of a neuron. And so I feel that to convince you that this is indeed the case, I have to show you some data. So what I'm going to do next is I'm going to talk about 3 different and seemingly unrelated neurophysiological facts that have been, I think, poorly explained or unexplained in neuroscience before, but naturally fall out of this framework. And then you will see how valuable this hypothesis of a neuron being a direct data-driven controller is. Okay, so what are those 3 neurophysiological observations? They are the spike-timing-dependent plasticity, in particular, the so-called anti-causal part of that, the temporal extent and the adaptation of the spike-triggered average and the spike history-dependent filter in neurons, and finally, the ubiquity of noise and variability in the brain, which naturally, all of which naturally follow from this controller framework. Okay, so moving on to the first observations, which is spike-timing-dependent plasticity, I should also introduce to you what this direct data-driven control framework really is. And for that, I, of course, chose the simplest, most basic case of scalar dynamic, xt plus 1 equals axt plus but, where x is simply a scalar that varies according to this equation, where a is another scalar and can be greater than 1. And in the presence of noise, it could breed instability. And that instability has to be suppressed by the control signal U, which is coupled through B. And the goal of the neuron here is to compute a control law, which is U out of X. And I call the coefficient proportionality W in this case instead of K before, because it is naturally associated with the weight of a synapse that connects this dynamical system to the neuron under consideration. So we would like to compute what the W should be to stabilize this dynamical system. Of course, you all know how to do this, and the dynamical system this simple can be stabilized in one time step in LQR kind of fashion. It's the simplest version of LQR where control has 0 cost. We're going to postulate that we would like to bring xt plus 1 to 0 to solve this one-step LQR problem. And by substituting the expression for U in here, we get that xt plus 1 is a plus bw times xt. To make sure that xt plus 1 will be 0 and the system is stabilized, we of course need to have a plus bw 0. So it doesn't depend on xt, and that means that w is minus a over b. So this is all very basic stuff. And of course, you can view this as a limiting case of a one-step LQR, but this is kind of natural thing that one would do to stabilize the system. But as I told you before, the question is, how can a neuron do this? Because the neuron doesn't know A or B, and so how is it going to compute W? And that's exactly where direct data-driven control framework comes in, and I'm going to explain it to you now in 2 ways. first geometrical to build up some intuition, and then algebraic. Okay, so to explain to you what is happening here geometrically, let's think about this triplet of numbers, xt plus 1, xt, and ut, as a vector in a three-dimensional space. Of course, because of this dynamics equation, these vectors don't live anywhere in this space, but they are confined to the subspace, which is in this case is this blue plane, okay, where all of those legit triplets, xt plus 1, xt, and ut, must live. Okay. Now, what will we need to shut down xt plus 1? The next step to bring it to 0, we need to bring this system's vector into the plane of xt plus 1, shown here in gray. Because the vector has to also live in the blue plane, it would have to be this red vector, okay? And how do we do this? Well, we can measure xt, we being the neuron controller, measures xt, then it builds up the orthogonal line to the red vector and projects to ut, and that's the control signal that the controller should use to obtain 0 xt plus 1. Of course, to be able to perform this calculation, we need to have this red line. And to have the red line, we need to know where the blue plane is. But if we already observed several triplets of xt plus 1, xt, and ut, we're going to have several blue vectors that run in, that live in this plane, and so we can use them to construct the blue plane, find the intersection with the gray plane, and compute the red line, which we will use to compute control. So that's the basic idea of direct data-driven control. Perhaps it sounds a little bit strange. Why would you do this way? And this is all kind of trivial, but you will see the power of the approach in a couple of minutes. Now I'm going to tell you exactly the same story, but using linear algebra. So what I just told you in linear algebra can be expressed in the following way, where this three-dimensional vector, xt plus 1, xt, ut, can be written down as a linear combination of previously seen triplets from the same system, from the same controller, which are all listed here, and they're weighted by this weights called G1 to GK, which is basically gives you a representation of that vector xt plus 1, xt, ut in the frame defined by those previously seen triplets. Okay, this is all obvious stuff, right? You can represent any vector in the subspace as a linear superposition of the complete or over complete basis over complete frame. Okay, so it seems rather trivial. And why is it even called the Williams et al.'s fundamental lemma? And that's because this isn't actually always true. And you've probably already seen this. And there is a prerequisite for this to actually take place, and that is the collection of previously seen vectors has to be sufficiently diverse to span the two-dimensional subspace. Because imagine that all the previously seen vectors are collinear with each other, like they're all like this one blue vector, but of different magnitude, okay? Then you cannot use them to define the plane uniquely, right? Any kind of flopping plane that passes through that line would do. And that would not define the red line correctly, and then you wouldn't be able to perform control. So that's why it's called the lemma, and it says under the condition that those vectors are sufficiently diverse, and they call this condition persistence of excitation, then you can actually perform this linear decomposition. Mathematically, this condition can be expressed in a variety of ways. It's convenient for us to say it as a matrix of the last two rows, submatrix of the last two rows has full row rank, or in other words, those vectors are linearly independent. So that's sufficient for the blue vectors to define the blue plane uniquely and to find the intersection with the red plane and to determine the control law, which maps from xt to ut that takes xt plus 1 to 0. So that's how simple the direct data-driven control is. It maps directly from the inputs to the outputs and bypasses even representing these parameters A and B. Okay, now that you're all experts in data-driven control, we can finally solve this problem and stabilize the system by setting xt plus 1 to 0. And to do that, we substitute actually xt plus 1 equals 0 into this equation. And now we have to solve it for ut, right? So the neuron controller observes xt. Its desired xt plus 1 is 0. Those two are fixed. But we need to find ut. Ut is a variable that has to be found by solving this equation, where this is a matrix of previously seen data, and g are the coefficients that are actually also unknown. So what we're going to do is we're going to solve for this coefficients g from the first two equations, and then plug in those g's in the last equation and find ut. There are 2 points that I need to make here. First, of course, there could be more previously seen triplets than 2. So this is an under constraint problem, and we are going to choose the solution that minimizes the norm of G. And second, you will see that the solution that I show you now actually doesn't require you to store this individual trajectories, which is crucial to make the solution biologically plausible. So without going through the calculations, I'm sure you can write down a pseudo inverse on your own to solve for G, and then plug in that into the expression for U. This is the part of W that the only part that depends on U. The W, which is the coefficient of proportionality, remember, between xt and ut, has this 2 sums in it, okay, that come from this solving this equation with a pseudo inverse. And these are basically covariances between x tau and u tau over history. So these come from this previously seen data points. And I want to emphasize that even though x tau and u tau have the same indices, because u is computed causally from x, there must be a little bit of delay. So what we have here is covariance of this neuronal activity u with the recently happening activity in the presynaptic neuron x. That's the first term. And the second term is the covariance between the, again, activity of our considered neuron u and the activity of the presynaptic neuron x tau plus 1 following u. So that's this anticausal term. And beta is basically a covariance of the first 2 rows, and it is generically less than 1. So already I hinted, of course, that this expression is going to be compared with spike-timing-dependent plasticity. Okay, if there is someone here who is new to neuroscience, I should tell you that this form of synaptic plasticity, what neuroscientists call learning rules for synapses, has been serendipitously discovered in the 90s. and it depends on the relative timing of the spikes in the neurons pre and postsynaptic to the synapse under study. And if the presynaptic spike precedes the postsynaptic, you get potentiation. And if it follows postsynaptic, then you get depression. And what I'm claiming is that this is paralleled by the equation for W we just derived from the direct data-driven controller, where the first term gets a non-zero contribution when there is a causal correlation between the presynaptic spike and postsynaptic. And the second sum gets a non-zero contribution when there is a correlation between a postsynaptic spike and a temporally following presynaptic spike, the so-called anti-causal part. And we can also get the switch in the sign from potentiation to depression, and the fact that the magnitude of depression is less than the magnitude of potentiation. So this, I think, already demonstrates the advantage of direct data-driven control as a model for a neuron, because previously the potentiation part could be naturally viewed as a extension of the Hebb rule to spike-timing-dependent plasticity, right? So pre-neuron causes spiking in post-neuron, so you strengthen the synapse. But it was very difficult to understand why, on the functional level, there should be this depression part in the anti-causal window, particularly considering that it's strongest when the delay goes to zero. Now, with the controller view of a neuron, this anti-causal part has a natural explanation. Because what does this mean, that presynaptic spike follows postsynaptic? Well, that spike could be causally related to the control signal, the output of the neuron UT, that affected the dynamics of the loop and perhaps led to the appearance of this presynaptic spike following the postsynaptic spike of the controller. So the existence of this loop effectively turns this window from anti-causal to causal, which physically, of course, makes a lot of sense. And that's why I think this is an important result that provides evidence that direct data-driven controller could serve as a good model of a neuron. Of course, there are many caveats here, and this is not the only form of STDP that has been observed, and different neurons could have different kinds of STDP curves. But also, I should say that the objective that we used here, which is nulling xt plus 1, is not the only objective that one could choose. And so I think that this is what's happening in biology, that different cell types have different objectives. And that's why they result in different STDP forms. But even for this STDP form, you can say, well, you only explained what's going to happen with the synapse when the spikes are shifted by 1 unit or immediately postsynaptic immediately following presynaptic. So these are just these two delta functions, okay? You haven't explained this whole temporal dependence. True, I did not, but in my defense, the dynamical system description that I used doesn't really have enough degrees of freedom to look at the temporal dynamics. This is a scalar equation that doesn't have any memory or inertia. And so there's no way that I was going to get all the swings out of it. So let me use this consideration as a motivation to move on and expand our modeling to try to include the temporal dependence and the higher order dynamics in the loop. So with that, I want to transition to the second biological fact or observation. And this will have to do with the neuronal response. But before I get to it, I want to say that, mathematically, what we're going to do, we're going to describe the state of the system not by a scalar x, but now by a vector x. And so A is now a matrix, and it describes the evolution of the state of the system, which can be influenced by the control. And because we still have one neuron, it's a scalar u times the vector b. And then it's observed by dimensionality reducing raw vector c, projecting onto a scalar y, which would be, say, the total postsynaptic current in the neuron. Okay, so this is now a slightly more complicated description of the dynamical system that the neuron has to deal with. So we're inching towards a more realistic description. But this already will have us, will keep us busy. So the goal as before is to find a controller, now back to the notation capital K, in terms of this inferred state x hat that would produce U. So I'm going to change tack a little bit here, and instead of worrying about K that much, I'm going to attack now the fact that we need to infer x hat. Remember, I told you that y cannot be used for control because many different state vectors x can correspond to the same scalar y. And so it's not a good dynamical variable. We need to infer x. So how are we going to do that? Or how a neuron can possibly do that? Well, actually, in this case, it's pretty straightforward. It turns out that all we have to do is to generate this a lag vector, as it is called, which uses the n, where n is the order of the dynamics, recent values of y. And this would be sufficient to reconstruct the state x if the system was autonomous and b was 0. Now, when we also have the control signal u, we also need to add n recent values of u to know that the current observations are not just autonomous, but could have been influenced by our control action u. So it turns out that this vector of two n variables, n recent observations and n recent controls of a neuron, is sufficient to describe the state of the system x hat. Now you must say, well, how did it happen that this kind of time delay embedding vector is exactly the state of the system? Well, in this case, I should remind you that for partially observed dynamical systems like this, they're only identifiable up to a linear transformation, because see, you can multiply both sides of the top equation by some matrix, and then you would get a different state representation x. Okay, so this x hat is just as good as any, and it's sufficient to uniquely represent the state of the system, and therefore to use it to compute the control signal. Okay, but this form of the inferred state has very interesting ramifications, because now K, okay, it would have to be a raw vector here because you need to produce a scalar control, has two parts. The part that multiplies the observations, which we'll call K feed forward, and the part that multiplies the previous control signals, which is this autoregressive loop that we will call K feedback. So in the linear approximation, the response of a neuron has these two temporal kernels that integrate previously seen inputs and outputs to compute the output of a neuron. And next, instead of computing the optimal K, which is what we did in the STDP part, I'm going to change the problem and instead compute those Ks from data. So what kind of data? Well, we're going to unhook our neuron from the loop and instead put it on the examination table and drive it with some Y and measure the outputs U. We will stack all those leg vectors together and also the output spike train, and we will solve this linear regression by pseudo-inverse and compute those kernels K feed forward and K feed back. Of course, you probably realize by now that what I'm telling you, you knew all along, that this, at least the feed-forward kernel, is nothing other than decorrelated spike-triggered average that neuroscientists have been computing forever, right? Which is basically, this is this covariance, right? That's the spike-triggered average part, and then this is the decorrelation part. And so you take the waveforms of the injected current, for example, preceding each spike, you average over them, and you may want to decorrelate by dividing by the covariance of the input, and you get temporal kernels like this. This is taken from mine and Sinovsky paper in the 90s, but many people have done this in all different contexts. And the point I want to emphasize here is that you really were computing those K feed forward kernels. And why should you care? Why should a control view be interesting to you? Well, first, because from the control perspective, now we know why these kernels had a finite temporal extent and the neuron does not respond instantaneously, right? That's because the dynamics of the loop is higher order. And to infer this dynamics, you need temporal history. And that's why you have those kernels. Also, you can see now that the kernels should adapt to the statistics of the inputs. That as a signal to noise ratio changes, the filter changes from more integrating one to more differentiating one. You can say, well, that's nothing new. That's what efficient coding theorists have been telling us. At high signal to noise ratio, take a derivative, low signal to noise ratio, integrate. Yes, that's true. They did. So why am I showing you this? Well, that's because K feed forward is only one part that determines the neuronal spiking. It has been known for a while to predict the spike timing correctly. You also need to include the spike history term, which I call the feedback filter, which depends on the previous spike timing. We computed that filter as well, and here are the shapes. Of course, there is a deep preceding the spike, and the textbooks say this is the refractory period because the sodium channels need to deactivate, so you can spike right after you just spike. Well, fine, but then there is this overshoot. Why is it there? And the shape of this filter adapts to the statistics of the inputs, just like the feed-forward one. So maybe just like for the feed-forward filter, where we had a functional explanation in terms of efficient coding, we should explain this adaptation also on the functional level. And this is where efficient coding has nothing to say, but the control theoretical view, from that perspective, K feed forward and K feed back, both part of optimal control. And so they should both be adapting to the statistics of the inputs. And so now, in theory, we should be able to explain the adaptation of those kernels in the feedback, just like we would have in the feedforward. And that's where an additional power of the control theoretic view lies. It also tells you what, the temporal extent of those filters tells you that, that's what you would have if you would have this feedback loop, right? The reason the neuronal spiking depends on the history of the previous spiking is because it's saying, wait, don't spike again, wait for that spike to traverse the loop and come back so that I can evaluate its effectiveness. And when it comes back, I have to make a call based on what it tells me, and that's what determines the shape of this filter, okay? Of course, none of this is the proof that our theory is correct. There never is, right? But this is another piece of evidence that suggests that the control theoretical view of a neuron is a useful and constructive one. So now that we were able to explain a couple of features of neuronal physiology, let me move on to the third one, which is the final one. And it actually comes from the same paper by Meinen and Chklovski. And this is probably the most famous figure from that paper, where they injected current waveforms like this. They also used them to, of course, infer the kernels that I showed to you. But the point of the paper was that the times of the spikes in the resultant spike trains were exactly the same up to a millisecond in each trial where the same current waveform was injected. So they injected the same current waveform in the same neuron, and they got the same spike trains down to a millisecond. Actually, this kind of result has been found before by Bryant and Segunda, so that's not entirely novel. But there is another figure in their paper, which is not as famous, but I think at least as or even more important. And that's the observation that if instead of a random waveform of current, they injected a constant current, okay, the square pulse, then the spike timing precision disappears. Of course, the spike timing precision doesn't come for free. So this precise spike time, temporally precise spike generation mechanism must be expensive. So when it fails, that means that there is something functionally wrong here. And that's why I can even call this a bug, right? So Meinen and Chklovski uncovered a bug in a neuron where they could make it generate an imprecise spike train by just injecting constant current. So this may seem like a little bit of a puzzle. Why would that be? But from the DDDC control perspective, this is a natural outcome. Because remember, I told you about this Williams fundamental lemma in the very beginning. And I said, well, for this direct data-driven control to work, the data matrices have to be full rank. But if the inputs Y are constants, then all of these columns have the same numbers in it. And this matrix is rank deficient. And under these conditions, the DDC cannot do systems identification, cannot find this plane. Remember, the plane can be flopping anywhere. It cannot compute precise control. Operationally, you could have seen this in the inversion of this covariance matrix, okay? If it's rank deficient, you can invert it. Of course, you can say, well, even though the nominally injected constant current, there must have been slight fluctuations, and then perhaps those were not reproducible from trial, and that led to the spike timing variability. But those fluctuations, of course, were present for the random waveform too. So why didn't they have effect here and they had effect there? Well, in the DDC framework, the answer is clear. This matrix may not be rank deficient in the presence of little noise, but it's ill-conditioned. And so small variations in the input, small fluctuations in the input lead to strong deviations in the output, which is the spike time. So it seems that the direct data-driven control theory is so powerful that it can capture not just the features of a neuron, but also its bugs. And to drive that point home, I want to show to you that it's not just the case when the input to a neuron is constant. It is also actually a case when the control law is constant. So remember how we computed this control law for W, right? In the first example of Spike-Timing-Dependent Plasticity, where it was just a scalar, and we said, well, the optimal value is minus A over B, and so we can compute it from the data. Well, that is actually true. But you know, once you converge on the constant W, in the online setting, in the closed loop, there is a problem, because those U are now WX's. And by being WX's, they make this matrix rank deficient. And going back to that Williams et al. Fundamental lemma, this breaks down the ability of DDC controller to learn the dynamics of the system. So if the external loop, or the internal loop around the neuron has constant dynamics, this would work just fine. But that is hard to expect in a biological system. It's probably changing with time. And so we need to preserve the ability for W to adapt. And the DC controller would not be able to do it in the form I described. How can we fix it? Well, there is a simple solution. Just add noise, okay? So what we're going to do is, instead of computing the control as WX, we add a little bit of noise. Not too much to deviate from the optimal control, but a little bit to make this matrix full rank. And I think then this becomes a viable controller, even for a time variant system. Where could this noise come from in the brain? Well, the obvious source is, of course, synaptic stochasticity. Perhaps you all know that in a central synapse, when a spike arrives at a presynaptic terminal, the probability of release is usually about 10%. So in 9 out of 10 cases, there is a silence, no EPSC on the postsynaptic side. Only in 1 out of 10 cases, there is a transmission. So there is a built-in stochasticity in every synapse, with some exceptions. There are, of course, many other examples of noise and variability in the brain. There is a very nice paper from C. elegans, where they found a neuron, and all neurons in C. elegans are known by name and are identifiable. So one of them was shown to serve as a noise injector in the circuit that is responsible for the stochasticity of behavior. Just like, as we said, you would need to. You may also know that in the birdsong system, there is a specific nucleus, LMAN, which is, I think, analogous to basal ganglia, that injects noise, which is required for learning. And also, of course, there are multiple escape behaviors, like the one in Drosophila, which are actually not stereotypical, but are somewhat stochastic. So there are multiple examples of variability and noise in the brain. There is also representational drift that we recently published on, that all kind of make sense from this direct data-driven control perspective. Okay, so I'm getting close to the end of the talk. I just want to address a few of the limitations of the model that I presented, because, for concreteness, I chose, of course, linear dynamics of the external environment. Of course, this is cannot be hoped for. The dynamics is going to be, of course, nonlinear. So how are we going to control it with the neuron? Well, nonlinear dynamics can be locally expanded as a linear. And so if you had a set of neurons, each of those acting DDDC in a particular part of the phase space, by approximating nonlinear dynamics as linear, we could have switch in linear dynamics here and implement control. So work in progress. And also I should address the issue, I said, okay, well, let's consider this one neuron against everyone else, okay? And it's a controller and trying to control the rest of the network, the rest of the neurons. But I could have applied this approach to every neuron in the brain. Every neuron thinks of itself as a controller. So each neuron is trying to control the rest of the neurons, and they all somehow have to come together and produce something useful. I don't know how that is going to pan out. And that, I think, is the million-dollar question. And that's what our future plans us to understand how those controllers coordinate together to produce something useful. Of course, there are many examples in science and life where this happens, such as national economies. They arise, at least in a free market system, from independent agents pursuing their own objectives. So I think there is hope, but we certainly cannot say that we know how this is going to be done. So to summarize, I suggested to model a neuron as a direct data-driven controller and presented 3 neurophysiological observations that would be a consequence of that hypothesis and are in fact observed, hoping to convince you that this approach is useful. And finally, I want to thank the people who were directly involved in this work, like Jason and Alex, who helped me with the data analysis, Anirvan and the rest of the crew with whom I had really illuminating discussions and who created the fantastic intellectual environment that made this work possible. Thank you very much.